So all cells go through the cell cycle, but not all cells actively divide all of the time. In fact, cells divide differently depending on what type of cell they are. So the frequency of cell division, so how often they divide, depends. Some cells divide more frequently, which would be things like types of cells that get damaged, so things like your skin or the lining of your digestive system. So your cells inside the lining of your stomach that are constantly being damaged by acid in your stomach are constantly dividing and repairing the stomach lining. Some cells divide less, right? Your heart muscle, once damaged, doesn't repair itself. So once your heart is adult-sized and formed, it really does not divide into new heart muscle cells. The same thing goes for your central nervous system, your brain and spinal cord. Once those are formed, they really don't divide anymore. That's why damage to the spinal nerves are so irreparable. So these differences result from what we call regulation. So these are going to be other ways that the cell controls, do I divide or do I not divide? Am I turning mitosis on? Am I turning mitosis off? Regulation is normal, and having regulation keeps our cells doing what they're supposed to be doing. When that regulation fails, so when those cells escape the typical regulation, that's when we end up with cells that are dividing when they should not be dividing, and that's how we get cancer and tumors. So here's a representation of the cell cycle as kind of a clock, and you can see we've got interphase out here with G1, S, and G2, and then we've got the mitotic phase here with mitosis and cytokinesis. And there are three main checkpoints that are highly important to the cell cycle control. The first one is the G1 checkpoint. This is usually the first checkpoint to ask, is the cell even going to divide? So if we say yes, we're going to go ahead and move on towards synthesis and go ahead and copy the DNA to get prepared to divide. Most cells that pass the G1 checkpoint do end up fully dividing into two cells. Cells that don't typically divide, like your brain, nerves, and the muscles in your heart, will actually say no here, and they don't go through the G1 checkpoint. What they end up doing is they go into a kind of stasis called G0 or G0, where those cells are in a non-dividing kind of eternal interphase, where they're just going to do their job and they're not going to worry about dividing ever. The next one that you should be aware of is the G2 checkpoint. This is kind of a final check right before we get into mitosis. We're going to check to make sure that we copied the DNA correctly. Did we copy using the correct formula in A's paired with T's and G's paired with C's? This is where we're going to check for mistakes, and we're going to go fix those mistakes so that we don't have mutations. This is where we're going to check to make sure that we have enough organelles, and this is just a final check to make sure that when we move into mitosis that we're going to be able to be successful. And then the last one here is the M checkpoint, and M is for metaphase. And so this is going to ask, are the chromosomes attached to the spindles? So in order to correctly use the spindle fibers, we need to make sure that the spindle fibers from both sides of the cell are fully attached to the chromosomes in metaphase. The chromosomes are in the middle, and then when we move into anaphase, we're going to actually pull them apart. If metaphase checkpoint is not correctly checked, we could end up with not actually breaking the chromosomes apart and end up with too many chromosomes in one of the two daughter cells. Now, we obviously don't have little workers running around the cells in hard hats checking all of the DNA to make sure that it's correct. Instead, what we have are proteins. So the proteins that are involved in, specifically, this is the G2 checkpoint here, are called cyclins and cyclin-dependent kinases. So a cyclin-dependent kinase is a protein that persists throughout all of interphase. So it's this little orange one here. And what you can see is that it's got a place to bind with another protein. Throughout interphase, this other protein called a cyclin builds up. So the frequency of cyclins is not enough over here, and then the number of cyclins increases as we get through interphase. And then eventually, by the time we reach the G2 checkpoint, we have enough cyclin to bind with the cyclin-dependent kinases, and they create this complex here, which is the maturation promoting factor. Once we have enough of those complexes, it's going to say, yes, you passed the G2 checkpoint, and you can go ahead and move into mitosis. For most cells, that first checkpoint, the G1, is the most important. If they pass the G1, they're going to go ahead and divide, and they usually are able to complete the synthesis, G2, rest of the interphase, as well as all of the mitosis and cytokinesis, and then go ahead and divide into 2 cells. When they do not pass G1, the cells do not divide. So here we can see this one said, no, we're not going to move forward with cell division, and it's moving into this kind of stasis phase here called G0, where it's going to continue to function as a cell, and it's going to continue to stay as if it's an interphase, but it's never going to progress through to the S phase of interphase. It's basically just going to stay in G1 or G0 the whole time. So we've seen a few examples of internal factors. Internal factors are just anything that's actually inside of the cell that helps to control the regulation. One of those was the cyclins and the cyclin-dependent kinases that form together into that maturation promoting factor. So that's an internal mechanism to determine if we're going through with the G2 checkpoint. Another one here is to properly attach the chromosomes at the metaphase plate. So that's going to be associated with the M checkpoint at metaphase. So internal factors are going to be any of these examples where the check is happening inside of the cell that's actively dividing. There are also external factors, so things from outside of the cell, usually its environment or its interaction with other cells, that's going to determine whether it should divide or should not divide. One example of those are growth factors. So growth factors are going to be hormones or proteins released by other cells that stimulate nearby cells to divide. So one example of growth factors is if you cut your hand, what will happen is platelets are pieces of blood fragments that are going to come to that area, and they actually help the blood clot so that you don't bleed. And the platelets will also release growth factors. And so the clot stops you from bleeding, but it also tells the cells around the cut that they need to go ahead and start doing mitosis to go ahead and heal that cut. And so that is an example of growth factors being used in cell repair to drive mitosis. Density-dependent inhibition. So density is the number per space, right? High density means we have a lot of things in a small space. Low density would mean we don't have very many things in some space. So inhibition is going to turn mitosis off compared to growth factors, which we're going to turn mitosis on. So basically what happens is when the density is high, when things are crowded, cells will bump into each other. So for example, when we're healing our cut, once our new cells touch each other, well, then they know that they've healed the cut. And so they will send signals to each other to go ahead and stop dividing because we don't need any more. We've repaired the damage. Most cells are also required to be anchored to something. So they have anchorage dependence, which means they have to be attached to other cells or some kind of substratum. Cells don't just float around in the body, and if they were to break off and move around in the body, they don't divide. So here's an example of a few of those external cells. So this is a petri dish. So this would be grown in a lab with a surface. So we can see anchorage dependent. The cells in here are only going to grow on the surface. You can see them growing on the bottom here. The ones that were kind of floating around up in this medium, those ones don't grow. Only the ones on the bottom are going to be able to grow. They form a single layer. This is a normal, healthy cell. And then what happened is the scientists reach in here with a scalpel blade and they caused some damage. So this would be equivalent to you kind of cutting yourself and causing some damage to your skin cells. In a human, platelets would come by and platelets would produce growth factors which would stimulate this to grow. This is obviously in a lab environment where the cells are stimulated to grow anyway. And so the cells fill in the gap. And as soon as the density-dependent inhibition kicks in, as soon as they're touching each other, they stop growth and they stay in that single normal one-cell layer. Cancer cells are cells that can break some of these rules. So they either have mutations in their control mechanisms, or they have other mechanisms of breaking some of these internal or external regulatory factors. So for example, in this one, the cancer cells are not growing in a single layer. You can see them growing on top of each other. And you can see microscopically that the structure of these cells is very different. Cancer cells form tumors because they continue to do mitosis even when they were supposed to have stopped. So they form tumors, and those tumors take up place in our body. They take up nutrients from us. Sometimes they move around in the body, and then they can cause massive systemic damage depending on what kind of cells they are and where they are in the body. So cancer is any type of cell that doesn't respond to the body's cell cycle control mechanisms. And they can do this in a lot of varieties of ways. And when we think about cancer, a lot of people talk about how, oh, I had breast cancer or pancreatic cancer or blood cancer or all sorts of different cancers. And we name cancers based on where the cancer cells first formed. So colon cancer happens in the colon. But cancer is a very generic term for just cells that are dividing when they should not be dividing. And so there's no one cure for cancer because all of the different cancers are formed from various different ways of breaking cell cycle controls. Even people who have different types of skin cancer are all formed from different mutations or different problems with their cell cycles. So some types of cancers can make their own growth factors. So instead of having to have an outside signal that says you can go ahead and grow now, they can tell themselves that they can do mitosis even though they shouldn't be doing mitosis. Some cancers can get a growth factor signal without actually needing growth factors. So they can receive the signal that they're supposed to be doing mitosis without actually getting that signal. And so they go ahead and do mitosis even though there's no actual growth factors present. And then many of them just have abnormal cell cycle control systems. So they might have mutations in their genetics that allow them to surpass the G1, G2, or M checkpoints. They might have ways of getting around the anchorage dependence or the density dependence. Any kind of mutation in any of those genes would allow them to go ahead and do mitosis when they're not supposed to be doing mitosis. A benign tumor is a lump of cells that is going to stay at the original site. So this means that it hasn't moved around in the body. It doesn't mean it's not dangerous. Most of the time, if you go to an oncologist and they say, oh, you have a benign tumor, what they're going to do is they're going to do a surgery to remove that tumor, and then usually chemotherapy afterward to make sure that they got all of it. Because if they leave even a single cell of that original tumor, it's going to go ahead and reform into a new tumor. A malignant tumor, mal usually means bad, is going to invade surrounding tissues. This could be done in a couple of ways. This could just be that the tumor grows so large that it starts to move into different tissues. So this could be a breast cancer tumor that starts in the breast and is able to progress into the muscle behind, like the pectoral muscles behind the breast. That would be malignant, even though it didn't actually break apart and move around. A malignant tumor can also form through metastasis. So this is when a piece of the tumor actually breaks off and then moves to a different portion of the body. This is really common in things like pancreatic cancer, lymphatic cancer, where they're spread throughout the whole body. And this is going to form additional tumors or mets throughout the body in other places. Once a tumor has metastasized, it's usually hard to get it out with surgery, depending on how many there are. So somebody might do surgery if you have metastasized tumor, they might not. Usually you're going to go through radiation or chemotherapy to try to shrink the tumors. If a tumor is in just one location, sometimes it can be radiated. Radiation, all of our cancer treatments are very damaging to the human body as well as to the cancer cells. And so radiation is just high energy damage, which is going to try to be localized on the specific cancer cells to kill them so that the body can kind of break that tumor down and heal itself. When this is a benign tumor in one spot, maybe if it's in like the leg or the arm and it's not in kind of the center of the body, this might be something good. However, you can't irradiate your whole body. That's really bad. And you can't radiate your brain. You wouldn't be able to do this for a brain tumor because it would damage the brain. So instead of that, for most metastatic cancers or cancers that are in sensitive areas, we use chemotherapies. There's tons of different chemotherapies. We've got tons of different drugs that we use to fight different types of cancers. A chemotherapy is a drug, it's a poison that targets the cell cycle. And so chemotherapies are going to target cells that are doing mitosis. So the goal with chemotherapy is for it to kill the cancer cells before it kills your body cells. People who go through chemotherapies, most of us have probably met somebody who's gone through chemotherapy. There's a lot of side effects. So hair loss, nausea, vomiting, usually weight loss with the nausea and vomiting. A lot of these symptoms come from the fact that the chemotherapy is killing cells that do mitosis. So your hair is constantly growing. The hair on your head is always doing mitosis. So the chemotherapy actually targets the cells in your hair follicles. The lining of your stomach, again, is constantly dividing. And so the chemotherapy actually targets the digestive system. And that's going to cause the nausea, vomiting issues. And sometimes chemotherapies work and they're able to shrink tumors and put people into remission. And then sometimes the chemotherapies don't work and they're not able to shrink the cancer tumors. Chemotherapy is a very broad spectrum cure, so it's not a cure, it's just a treatment where we're trying to kind of kill the cancer before it kills you. The recent advances in cancer research are trying to find more personalized ways to treat cancer. So if you can figure out exactly what the DNA sequence is of a particular tumor, you might be able to create more of a targeted therapy for that specific type of DNA mutation or cell that has become cancerous. Obviously, this would be great. However, it leads to some other issues that, if you have cancer, you need to be able to get healthcare and find healthcare that's good enough to figure out exactly what type of cancer you have and what caused it, as opposed to just kind of treating it as a blanket cure. So this is the kind of thing that's going to be more accessible to people who have advantages and privilege and better healthcare, where somebody who isn't able to get healthcare, is going to a free clinic, is probably not going to have access to these more experimental, newer, more personalized kinds of treatments.